I'm joined today by Dr. Alison Eenennaam, who is a professor of cooperative extension in animal genomics and biotechnology at the University of California, Davis for the past 20 years. Her research focuses on the use of DNA-based biotechnologies in beef cattle production and agricultural systems. She's served on national committees such as the USDA National Advisory Committee on Biotechnology in the 21st Century Agriculture, and has received numerous awards, including the 2014 Borlaug Cast Communication Award and the National Award in Animal Breeding in Genetics in 2019. And in addition to that, Alison is also a member of our GEN Biotechnology editorial board. So welcome, Alison. How are you today? I'm so excited to have our chat.
Yeah, well, thanks, Faye. I'm excited to chat with you and get to know you a little bit better too.
Absolutely. Now, I'm really excited because on our April issue of GEN Biotechnology, which I have the cover here, we were so excited to feature your paper, which was titled Animal Health and Food Safety Analysis of 6 Offspring of a Genome-Edited Hornless Bull, featured on the cover here. So I wanted to start our discussion about this paper that you published with us in April. Can you give our audience a quick summary of this article, and then we'll dive a bit more into the 2 cattle that are featured on the cover here?
Okay, sure. So basically, this is kind of a long saga. So there was a company called Recombinetics. There still is a company called Recombinetics who has a division of Excelligen that produced 2 genome-edited bulls that had a targeted gene knock-in to introduce a variant of a gene, so an allele, from a different breed of cattle. So beef cattle don't grow horns and dairy cattle do. And so normally farmers remove horns from dairy cattle to protect other animals from getting skewered by the horns and to protect human workers. But there's this allele that exists in beef cattle like Angus that is actually a naturally hornless allele. And so this company introduced that allele into dairy cattle background germplasm and produced these 2 bulls that were homozygous for the allele to not grow horns. It's an allele that's called polled. And so they were working with us. They produced the animals and then we brought them to Davis, and we were interested in making sure that that trait was transmitted faithfully to the next generation. And so we bred some of our cows here at Davis with those bulls and produced offspring. And in fact, because hornlessness or polled is a dominant trait, they didn't grow horns. So that was a pretty easy phenotype to see. They kept not growing horns every day. And we raised them here and basically were able to show that they were just normal cows, or in the case of bulls in the case of the boys, cows in the case of the girl. And basically what that paper did is a really thorough analysis of the milk and meat composition and the health status of those 6 offspring. And the reason we did that was because at the moment, if you do genome editing in food animals in the United States, they're under the FDA's regulatory purview, and they're actually regulated as drugs, new animal drugs. And part of getting a drug approval is to show that you have the milk and meat from the animals you're working with is safe for human consumption. And so it's a pretty deep dive into the kind of the compositional analysis of the meat and milk from those animals, in addition to their health attributes. And that was over a period of several years that we've been working on this. And so that's really the culmination of an end of a project that started in almost 5 years ago now. And so it's nice to see it in press.
Absolutely. And I think livestock biotechnology is such an important applicable aspect of the field that has such a close tie to our day-to-day lives, whether it be the food and safety of the milk and meat that we consume. And I personally am not a livestock biotechnologist. Could you explain to our audience, what is it about having the hornless bull that is significant as far as improving the way that we approach food?
So in this particular case, the alteration that was being made was to address an animal welfare concern. So when farmers remove the horns physically, they usually either burn it off at a young age or apply a caustic paste to basically kill the horn bud cells before the horns form. And that's painful for the animal. Farmers don't enjoy it very much either. And it's just, I guess I'm a geneticist, I have my own biases, but it's just better to remove them genetically, and then you don't actually have to physically do this procedure. But more generally, I think there are traits or characteristics we can introduce into our animal breeding programs using technologies like genome editing that really are going to help with the overall sustainability or reducing the kind of the environmental footprint of food production. And particularly, I think one that's of importance here is disease resistance. And so we lose about 20% of animal production to disease. And so if there are edits that can be made that can render an animal no longer susceptible to certain diseases, that would be very compatible with kind of an interest in trying to reduce the environmental footprint of animal source food. And a really well-known example of that is actually in the swine or the pig industry, where a knockout in a particular gene results in pigs that are no longer susceptible to a really nasty virus that's called the porcine respiratory and reproductive syndrome virus, PRRS, which basically is, as it sounds, it's a respiratory virus. You might be a little familiar with those at the moment, and also affects reproduction. And so those pigs appear to be no longer susceptible to that virus. And so those are the types of traits that breeders would like to introduce into their breeding programs. And more generally, I think genetic improvement of both our plants and animals has been a huge contributor to reducing the environmental footprint of food production. If you look at the yield of corn today or dairy cow, they're way more productive, and that basically reduces the you can grow more corn on an acre, you can produce more milk in a cow, then you need less cows to produce the same amount of milk, and similarly with the plants. And so it really is a big part of the sustainability story when you can have improved genetics.
Yeah, I love how you're highlighting all of these really important applications that these technologies can achieve, from disease resistance to reducing our environmental footprint. And I think I want to shift a bit deeper into that. Now, I know in, for example, the bigger picture section of your paper in GEN Biotechnology, you mentioned that the findings in your Hornless Bowl paper supported this proposal that product compositional data from genome-edited animals should only be required by regulatory agencies if a hazard has been identified. So I was wondering if you can speak more about the current state of livestock regulation and any obstacles that prevent these technologies from really reaching their full potential that you're describing.
Well, you might have asked for a 1-hour podcast if you want a response to that. So I will give you the abbreviated form, and something as you mentioned at the outset, I've worked in this field for 20 years. And basically, when genetic engineering first started, and this is back in when it was GMOs, and we were introducing entirely novel transgenes that maybe came from a different kingdom into plants and animals, there was kind of regulation set up with this relatively unknown technology to make sure that there wasn't the introduction of a novel allergen or a novel toxin. So what they were looking for there is, did you bring in a protein that's now being expressed that is going to create a food safety hazard? That was one of the major questions. And you mean conceptually, if you think about it, yeah, you could do that. Like you could bring in the toxin from puffer fish and put it in cows and make toxic milk, right? It would be conceptually possible to do that, but you would ideally look at the protein and determine its allergenicity and its toxicity to make sure that you weren't doing that before you would go to the effort of producing it. But irrespective, that was an important part of the safety evaluation. Let's fast forward 20 years now, and we've got new technologies like editing, where we're not introducing a foreign transgene necessarily, like you could, but not necessarily. And more commonly, you're doing a targeted gene knockout, where you're not actually introducing any novel DNA, you're just inactivating a gene like the virus receptor, for example. And so there is no novel protein to be allergenic or toxic in the meat and milk. And what's being asked is, well, you still need to show what's called substantial equivalence in terms of the milk and meat, in the same way that this was requested of transgenic plants back in 20 years ago. And it's not inexpensive to do the type of data analysis that we did for the paper that's published in GEN Biotech. And just to give you a feel, that particular project was funded by the USDA through what's called a biotech risk assessment grant. And that was a $500,000 grant, not only to raise those animals for their lifetime, but also to basically collect the milk and meat and analyze them for approximate analysis and minerals and vitamins and all the rest of the nutrients that make up animal source foods, but also the controls that we brought along with them that were not genome edited to be contemporary comparators and what have you. So there was quite a barnyard involved in that whole experiment over 4 years, basically. So we had to have the cow and a control, and that actually, you asked about the cover. So that cover shows Princess, who is the one female offspring that we produced in our experiment of the 6. So we had one female, and alongside her is a horned Hereford control, born at the same time. And so that would be an animal that we would compare the data to. But what's kind of difficult to put into an experimental design is when you don't have a hypothesis you're testing, so there's no protein to determine if it's toxic or allergenic. You just have compositional data, and we drink milk with different compositions all the time. So, Jersey milk is different to Holstein milk, and milk from a cow that just gave birth is really different to a cow that's 20 days in milk or 100 days in milk. And that's part of the natural variation of our diet. And it's not a food safety hazard. It's just compositional variation. And when you start asking for that data, then it becomes incredibly expensive, and it becomes often beyond the realm of certainly a research budget of university animals. And in my case, I was able to get a grant, but I doubt I'd ever get another grant to show that horned cows' milk and meat is no different to animals that don't have horns. That's not going to happen again. And that's part of the reason we wanted to do this, was we wanted to do this work to have scientific data to back up the argument that if there's no novel hazard, that is, there's no new transgene or exogenous DNA, then requiring compositional data doesn't make sense when there's no, obvious thing to even test when you're doing these. What are we looking for? Changes in vitamins, minerals, protein, proximate analysis, what is it? And if you can't name what that is, you can't really design experiments appropriately to power them to look for that change. And so it feels a little bit like a fishing expedition, because you don't have a hypothesis, and it's not very, I guess, kindly put, it's not the most interesting work for a research lab. But certainly I couldn't find a student that put their hand up for it. So my lab manager and I just did the paper ourselves, because it was kind of, well, what was your PhD on? Oh, I wanted to see if milk and meat was different from, cows with horns. It's like, okay. So that was the rationale behind it. And so I think it is important in setting as part of this regulatory discussion. And that's where I think that it's important to have these types of discussions to ensure we're not asking questions that just make the whole process more expensive, that don't actually return any benefit to public safety. I think regulations should be based on risk, and very risky things should be regulated very highly, and very non-risky things should have a very low bar. And that's what should trigger it, not just the fact you happen to use one particular molecular biology technique to produce your product.
Yeah, I think you highlight a really important point, that research takes time and money and resources to do. And sometimes with a lot of these obstacles, we ask the question, are we really being efficient in the way that we're using these resources? Are we really bringing these technologies in a way that are positively impacting the community, or are we really getting bogged down by all of these details? So I'm curious, now that this paper is published and out, are there next hoops to jump through before this technology with these genome-edited hornless bulls? Are there additional hoops that need to that we need to go through before these gene-edited livestock can really reach the applications that you described in the beginning of our chat?
So to answer the question about those specific bulls, so all of those bulls and their offspring and the offspring of their offspring have all at this stage been incinerated because they're unapproved animal drugs and they're not allowed to go into the food supply. So they were always a prototype. They were not elite genetics. They weren't the best looking bulls, quite frankly, and they were not the most optimal genetic bulls. They were proof of concept. And the proof of concept was, if you inherit this allele, it will produce a hornless animal, and it did. And we now have proof of concept that the milk and meat is no different. So what would happen next would be for a developer, and that's not a research scientist, so it would be a company like Excelligen, would work with people in the breeding industry, or companies in the breeding industry, most likely artificial insemination companies, to work with them to introduce those alterations or that allele into their elite genetics of the dairy bulls. And then that would be distributed through the AI, so artificial insemination, not artificial intelligence, to the dairy cattle population. And the next generation of calves resulting from those matings would all obviously not grow horns. And for that to happen, basically, there needs to be a path to market for these elite animals that would have this targeted insertion of the alleles that don't grow horns. And for that is kind of not really in practice at the moment because it is still a little bit unclear in terms of when you're doing a knock-in like this, if you have to go through the drug approval process, because that details quite an extensive multi-generational evaluation that's required, which would be, in the case of cattle, obviously a very long 10-year process or if they're going to do what they did recently for a couple of bulls that had knockouts, where the FDA exercised enforcement discretion and basically looked at the animals and said, these are really low risk. It's really not a good use of taxpayer money for us to enforce the requirement for drug approval for these low-risk animals. We're going to give enforcement discretion and allow these animals and their offspring into commerce or the food supply in the absence of a drug approval. So we're not approving it. We're just saying this is so low risk that it's really not worth our time to regulate it. And therefore, we're going to give it enforcement discretion. And so if that's a path to market for this technology in food animals, then maybe that's the way to go. And in that case, they did not require product composition from those animals because it was a knockout of a known allele that already exists in the cattle population. And so the company was not required to produce protein or food data in that case. So that's maybe a little bit of a hole in the or opening to the sky in terms of moving things forward. But that's not going to be true in other countries. So the EU, for example, considers all genome editing to be exactly analogous to traditional genetic engineering, and that has more or less precluded the use of that technology in the country, at least the growing of genetically engineered crops. They certainly import the heck out of them, but I guess that somehow is different. But it really does complicate it when it's not consistent regulations around the world. And it's ironic in America. So plants that are edited that could have been achieved using conventional breeding, like knockouts, are not treated differently to conventional breeding, but animals are. They're treated as new animal drugs. And so even within America, there's very different pathways for plant food versus animal food because of the different regulatory jurisdictions that plants and animals fall under.
Yeah, I think this could be a full separate episode, all of these different avenues of regulation, right? And I know for our April issue, you also wrote a great Views and News piece about a paper from the CRISPR journal that was working to CRISPR a gene for a specific cat allergen. So that's, again, Views and News in our April issue of GEN Biotechnology. I encourage everyone to check it out. And in that piece, there were also a lot of interesting discussion points about regulation when it comes to CRISPRing our pets. And I think you mentioned a few examples like glowfish and other types of animal regulation that is also present in society, and that there are all of these other ways in which I guess gene editing can take a part in our day-to-day lives, from food to pets. So I was wondering, on that angle, did you have anything in addition to say about the pet angle? And if it is hypoallergenic cats, what is the regulatory status on that side of animal science?
So, well, I think people should really look at that episode, or look at that journal article, because there's a picture of my cat there. So I was very excited to co-author a paper with my cat. But the example you use, which is glowfish, is a fluorescent aquarium fish that I have in my house. And that is actually a genetically engineered transgenic. So it expresses a protein from a jellyfish. That's what makes it glow. And there's orange ones and green ones and blue ones. They're really pretty. I really like them. But they are under the same enforcement discretion with the FDA that I mentioned earlier, with the two knockout bulls, the slick bulls that were given enforcement discretion. So they are not, they're selling an edited, or in this case, a genome engineered animal that hasn't been approved, but it's been given enforcement discretion, which means it's a low risk product and it's not a food, or maybe at some frat parties, but for the most part, it's not a food. And it is therefore allowed to enter commerce in the US under the way it's regulated. However, for other animals, it would be on a case-by-case basis as to whether you would get enforcement discretion. And one of the things they look at is whether or not there's a likelihood that that animal could spread its genetics beyond the aquarium tank in the case of the glowfish. And glowfish can't survive in normal temperature waters in America. So they're very low risk in that regard. Whereas if you did CRISPR a hypoallergenic cat, so you knocked out the gene that results in the allergens that people respond to, so that no longer produces that protein, then those cats would obviously have that characteristic. And then would that characteristic be able to be spread beyond, your house cat? And I think the answer there is very clearly yes, because there's feral cats around, and cats get out and about, as you probably know if you have your windows open at night and you hear the fuss going on. And so that would come up as, because it's editing in an animal, it would be legally under the FDA's authority. And so they would have to consider that case as to whether or not that would be enforcement discretion, which is what the glowfish has, or if they would want a full animal drug approval. And part of that would be getting the environmental impact assessment as to whether or not this trait of being not allergenic would be something that might spread in the feral cat population, which just opens up a whole bunch of interesting questions that I'm glad I don't have to do the research for, because I couldn't imagine how you would go about doing that. But it does kind of highlight the fact that, and actually one of the other things that has been in the history of pets is that there was some other groups that said they bred hypoallergenic cats, and in that case, they wouldn't have to jump through any hoops, because conventional breeding is not regulated. And so the method you use to produce your hypoallergenic cat can really trigger what sort of regulatory pathway you go down, even though at the end of the day, they might be exactly identical animals, similar to our hornless animals that are, they have the same allele as naturally occurring beef cattle. But because it was introduced using genome editing, it triggers a whole different regulatory pathway. And it's a little bit inconsistent in terms of the risk. So what is the risk? Because surely that exists in conventional breeding. And if it's not regulated in conventional breeding, why would you regulate it when you're doing it using a different method such as CRISPR? And that, I think, is a contradiction that needs to be addressed, because you shouldn't have different regulatory bars for the same product.
Yeah, that's super interesting that depending on your method, that there are going to be different as you said, different regulatory paths for the same product. And like I said, I think we could have a whole separate episode on just digging through all of these.
I never wanted to become this familiar with regulatory issues. Trust me. It's like, I'm not a regulatory scientist. I'm an academic scientist. And it's just, unfortunately, or maybe fortunately, I mean, this is the time to have these discussions. So this technology is it's not really that new anymore, but it's a technology that has so much potential for use in agriculture. And I believe we got the regulation of genome editing, engineering really wrong. So it made it so expensive that the only people that could use it was large multinational companies in large acreage crops. And it was set a really bad precedent because it became conflated with these big companies rather than being associated with the academics that created the technology that were using it for all sorts of really useful things that then they could never bring to market. And I fear that, CRISPR was developed by public sector scientists, and you often hear it democratized the technology. Well, if you now introduce regulations that make it, a multi-million-dollar effort to get anything through to commercialization, you've undemocratized CRISPR to some extent. And so I think regulation should be proportional to risk. I'm not saying just, throw the bucket out and we're done. But if it's a low risk product, then there should be a different pathway, I think, than a high risk product. And I think that looking at the characteristics of the product rather than how it was produced or what, it was zinc fingers or CRISPR or whatever, it doesn't really matter. It's like, well, what is the end product and what are the risks associated with it? And what are if there's no novel hazards, then don't ask it to do extra things that you don't ask of normal, breeding. And that's where I think if you make it really expensive to use this technology, you'll disincentivize its use and potentially stop a whole bunch of useful applications actually coming to market.
Absolutely. Absolutely. And I want to maybe, shift gears a little bit and talk about, first of all, your journey and your expertise. And it's evident that you've had exposure and your feet wet in all of these different aspects of animal science, from the research and also diving into discussions about the regulatory aspects. And I just wanted to ask you, what got you interested in this area of animal biotechnology and genomics that really sparked this journey for you?
Well, it's funny because I'm as old as dirt. And so I was born in 1963. So 10 years after the double helix paper, and quite a few years before biotechnology even existed. And so it wasn't as if when I was a kid, I said, oh, what I want to be when I grow up is an animal biotechnologist. It just wasn't even a field. And so I feel like I've kind of grown up with DNA and biotechnology. And so it's just always kind of a new thing. But I got interested in animal breeding. Well, I grew up in Australia, and of course, a lot of sheep down there, a lot of cattle down there, very agriculturally, we rode to prosperity on the sheep's back is kind of the common phrase. So a lot of familiarity with agriculture. And I really got interested in the work of, quite frankly, Norman Borlaug in terms of what he was able to do with genetics to help address food insecurity. But I was always interested in animals. And then I got an opportunity to work on a Santa Gertrudis embryo transfer ranch in Texas when I was going through agricultural science degree. And that really kind of opened up my eyes to, how you could use advanced reproductive technologies in cattle. And then I came back to Davis and I did a master's in Davis in animal genetics, and then worked in the field for a few years out with ranchers, and kind of got to understand agriculture, and then came back and did my PhD. And then I actually worked at Celgene here in Davis, which was just as it was releasing the Flavor Saver tomato, the first kind of genetically engineered food product. And it was a huge commercial success, or at least it was a huge public success here. It was really, you could go and buy it at the supermarket here, and it was well received. It was a tomato that basically didn't go rotten as quickly. And then I kind of watched the development of the anti-GMO narrative, and there was a particular guy called Jeremy Rifkin who really started the anti-BST, the recombinant bovine somatotropin kind of fake news, if you will, and created all sorts of fear around that technology. And that just kind of took off, and it actually spawned the university creating my position, which was to do public outreach on biotechnology and genomics around animal biotechnology. And so I owe my job, ironically enough, to kind of fear-mongering and anti-science sentiment. And it's been a really interesting juncture, because I'm 75% extension. So I work with producers, I work with the public on extending information from the university to those groups, and then 25% in a hardcore molecular research lab. And so I'm this really schizophrenic person that is like works in very different crowds and very different audiences, and perhaps doesn't feel 100% comfortable in either group, but really has a broad experience with the different issues that the public has on these technologies, and then also what farmers need and what problems they're trying to solve and how it all might get addressed by some of the modern biotechnologies.
Yeah, I think this outreach aspect is especially important, really in all fields of biotechnology, and helping people without the expertise really understand and be exposed to what these technologies are and how they can be really impactful, and not to be intimidated when you don't fully have that science background. So I'm curious, what are things that science communicators or people who do these outreach, what are things that we can do better to help promote these technologies in livestock?
I wish I had a good answer to that question. I think we've had one or now two approved genetically engineered animals. So obviously what we're doing is not working very well. But I think if you hear the science communication scientific community, their argument is that often scientists are wanting to educate people or give them more information, and I think it's called the information deficit model. And actually that's not what connects with public audiences particularly, and you want to lead rather than with technical details, with the why, what problem you're trying to solve, and try to connect it to whatever shared value that audience might have. And as I said, I work at a lot of different audiences, and so I'll have a very different kind of approach if I'm working with a group of agricultural producers than I might have with I don't know, PETA, or a group that's just non-GMO project or something, and so I think you need to be really thoughtful about your audience, and you can't just take the talk you gave at the Gordon conference and go and give it at the kindergarten. You need to think differently, and I think we don't do a very good job with teaching our scientists how to step back and think what's going to work well with this audience, and what is my shared value with this audience. Because if they don't trust that you've got their interests at heart, and often you'll go in with a title and you're coming from maybe an institute that they don't trust, you're not always going to have that trust on side. And I think we've seen during kind of the last couple of years with the misinformation around medical stuff like vaccines and COVID, that if you don't have trust, it doesn't matter what you're saying, and basically if you do have trust, a colleague of mine says, and it doesn't really matter what you're saying, which is true, but hopefully the scientists can get trust and then follow that up with actually actual truthful information, or at least evidence-based information. And so that's, I think, a lesson to learn, and one that doesn't flow naturally, because I am very evidence-based in the way I live my life, and my kids have had all their childhood vaccinations because the data clearly says that's the best thing for them. And as a parent, I'm going to use that as my decision-making criteria. But I understand that other people use different metrics for their decision-making criteria, and it's how to get to those audiences that aren't swayed by science, and we don't get there by screaming facts at them. And so how do you kind of work with, well, what shared values do we have, and can I actually communicate this information and in a way that resonates with them and doesn't alienate them from the get-go?
I think you captured so many important points, key ones being know your audience, establishing trust. Like at the end of the day, we're talking to humans, right? We're not talking to robots. And sometimes that really evidence data-based scientific drive may not be the best match for your appropriate audience. And keeping in mind that we are talking to human beings and not people who only look at numbers is just so important. I think sometimes, especially with people with a more hardcore science background, tend to forget when it comes to engaging with these outreach conversations. That's so important.
Most people don't make decisions based on the data. You you go with your gut on. I mean, certainly I do with, areas I don't know anything about. I'm going to, I kind of trust that guy, or, that girl, or, what my neighbor said. So, I do it in other areas, and I probably don't even know that I'm doing it. What is it? The chronic Dunning-Kruger effect. I probably don't even know that, but I do appreciate that you make these mental shortcuts, because you don't have time to do every single piece of research on every single thing, and it's frustrating when you're an expert in that area and other people are making decisions based on information that's being put out intentionally by groups to mislead them, and they're getting credibility from that audience that belongs rightfully to the incorrect information. And how do you get minds opened up to the fact that that's actually how it is? And I think that's proven very difficult. And certainly I think the scientific community has struggled with, particularly more recently, just with the broad reach of social media and the kind of tribalism that ensues there, in terms of who you follow on Facebook or Twitter or whatever, and you just kind of reinforce the messages that you agree with, and you don't ever expose yourself to contrary points of view. And that goes for all stripes of people, and it is quite dangerous, I think, because we don't actually have often discussions with people that have varying viewpoints to our own in a comfortable way anymore. It's just you're kind of either with me or against me, and it's really black and white. There's no shades of gray.
Yeah, and I think it just speaks to how important it is to have people in this outreach space, people who can bridge that gap between the experts and the broader audience, because there's so much room for miscommunication there, that when it does come to having these technologies reach their full potential, sometimes it is that communication gap that becomes a barrier. And so outreach is super important. Super excited that sounds like your role, what you say, 75% is outreach?
Mm-hmm. Yeah, extension is the kind of, it's been in the land grant universities for a million years. It used to be taking, seed varieties to the farmer, and you should try this seed variety, not that one, or whatever. And it's changed over time. And now 2% of our population is agriculturally working. And so 98% of people are not working in agriculture, and it used to be almost the other way around. And I personally feel like extension needs to shift its model to actually explaining agriculture to the 98% of people, because without just kind of a basic understanding of, why would you need insect protected crops? I don't want GMOs. I don't want insect protected crops. Well, if you don't know that insects eat the crops, and if there's no crops, there's nothing to eat, then, it might be quite a sensible thing to say, I don't want to have a GMO crop. But if you look at what the problem that's trying to be solved is, and have an understanding, well, alternatively, I could spray it with insecticide, which to me is not as good as having an insect protected GMO crop, then you don't know the real trade-offs that you're talking about, and what the actual implications might be to the farmer, or more generally global agriculture, if you say, no, you can't use that innovation, because there are very real costs to doing nothing and not using technologies. And I think that's often not elaborated, and without an understanding of kind of the problem, you really, you can make decisions that aren't actually the best, because the trade-off is not really obvious to you.
All right, looks like we're just about out of time for this episode. Alison, is there anything that you haven't yet shared in this discussion, whether it be about the regulation aspect, the outreach aspect, the science aspect, anything that you haven't mentioned yet to our audience that you'd like to just close us off with?
Well, I guess I am pretty passionate about science communication, and it really concerns me. And I think Carl Sagan had similar misgivings many years ago, that we live in a very highly technical society, and yet we seem to have less and less understanding of it. And it's I think super important for us, if we do want to have kind of social license to use some of these technologies, that we have to have not only scientific discussions, and that's what I like about GEN Biotech, is also the editorials and the comments that are just beyond the basic science, because it's more than just about the basic science. And so I think it's a really important aspect of the journal that we have the broader societal discussions around different topics.
Absolutely. And I just want to echo that sentiment. We're really excited at GEN Biotechnology to not only cover the original research, but also a great array of front matter from commentaries, perspectives, et cetera, about all of these different aspects of the biotech field that we discussed today with Alison. So please do check us out at genbiotechjournal.com.
Thanks, Faye.
Thanks, Alison. This has been such a great chat, and I'm excited to come back next time with more exciting guests.